in the last videos that we were doing mitosis, we were going from one cell to two cell. We were making identical copies. In this exercise, activity 3, we're going to go from one diploid cell. Helps if I spell it right. One diploid, which would be 2N, down to 4 haploid, 1N cells. So we're going to start off, we're going to do this with 2 chromosomes. So in this picture, I've already gone through interphase, I've already replicated and made my sisters. And then the first thing that we're going to do in meiosis 1 is we're going to go through our PMAT. So we wound up our chromosomes, and we're going to actually find our homologous chromosome. So mom and dad's chromosome 1 are going to find mom and dad's chromosome 2. The 2, so here we go. So here's chromosome 2, the long one, mom and dad's, and here's the short one. They found each other. They found their partners. Now in prophase 1, and that's what we're talking about here, prophase 1. So again, our nucleolus will break down, the nuclear envelope will start to break down. Okay, so I'm breaking it down. And we might have an event that is called crossover. Now crossover is where we're gonna kind of swap DNA on those homologous chromosomes. Remember I said it's kind of like they're going in for the smooch, and we swap some spit here. So we're going to have to exchange some of that DNA. Oops, so we don't want an unequal sharing. That would lead to a mutation. We don't want that now. So we're going to have a crossover at least with this. And again, remember this increases genetic variation, which helps survival of our species and makes more of us different, you can say. So after prophase, then, I don't have my centrioles in, so remember the centrioles, they replicated and they're pushing to the poles. They're going to continue on their journey, and then we're going to go right into, let me move this out of the way, metaphase 1. Now, metaphase 1 is a little different. You should be able to identify the difference between metaphase 1 and metaphase 2. 2 and I do 1. Okay, so right now I'm a mess, but in metaphase 1, this is where we have like our little square dance that I talked about. So find your partner. We're not going to go round and round, but we're going to line up. Okay, so we can line up this way, where we have mom's on one side, mom on the other side, and dad, they're opposite sides. Or we can line up the same way, and if you remember, that's called independent assortment. So that has to do with how they line up. So again, you can go here or here, it doesn't matter, or here, alright? So independent assortment, again, that increases genetic variation. So we're going to pull our chromosomes, our homologous chromosomes apart. I call it the great divorce. So we have our divorce, and they're going to pull to the opposite sides. And at that point, we've now entered anaphase 1. Once they get to their opposite sides, we're going to make our nuclear membrane. We're going to start forming that, and that would enter into telophase. And that, again, ends the nuclear division portion of this cell division. Now, at this point, we have taken our cell from 2N down to 1N. Notice we only have one short and one long, one short and one long. We don't have both. We don't have 2 shorts and 2 longs anymore. We've split up our homologous chromosomes. So we've gone from being diploid to haploid. All right, so next we're going to undergo cytokinesis, which will end up dividing the 2 cells, and then we're going to enter meiosis 2. All right, so let's pause so I can clean the board.